This is the recording for Chapter 7 on membrane structure and function. So this is going to focus on the plasma membrane of cells. And I'm going to skip through some of this beginning stuff. Okay, the plasma membrane is, we always use this term fluid mosaic. The plasma membrane is a fluid mosaic of lipids and proteins, okay? The main component of the plasma membrane is a double layer of phospholipids. We call it the phospholipid bilayer. And phospholipids have an end of that molecule that is insoluble in water or hydrophobic, and then an end that is soluble in water or hydrophilic. So we say that phospholipids are amphipathic because they have hydrophobic end and a hydrophilic end. So phospholipids are amphipathic, and so basically the head of the phospholipid, you can see phospholipids here, and they formed a double layer, and the tails are facing each other and the heads are facing away. So the heads are hydrophilic. They are composed of polar covalent molecules that are soluble in water. And so they face either outside the cell or inside the cell, into the watery environment, outside or inside of the cell. The tails that compose the phospholipid, so the phospholipid has a phosphate head, and then it has these two fatty acid tails, and those tails, since they're fatty acids, they are hydrophobic. They're nonpolar, which makes them hydrophobic or insoluble in water. So they face each other so that they don't contact the water inside and outside the cell. Okay. So the fluid mosaic model of membrane structure describes the large protein molecules that are embedded within the bilayer of phospholipids, and all of these molecules are able to move past each other, and it describes the protein molecules as bobbing, like you know, I don't know if you have ever bobbed for apples, because that's kind of an old-fashioned thing to do at Halloween. But bobbing for apples, when you put apples in like a bucket or a tub of water, they float and they bob up and down. They bob around as they're moving. So you can kind of picture the plasma membrane as the phospholipids are the fluid part, and then the proteins kind of bob around within the phospholipids. So that's what is meant by fluid mosaic model. Fluid means that this plasma membrane is a fluid. It is not a solid, and it's not like you see in this illustration. It looks like every molecule is stuck in one place. But these phospholipids can actually move and squeeze past one another. They don't always stay exactly in the same spot. And these proteins can also do the same thing. They can move around. They're like bobbing around in the phospholipid bilayer. You can kind of see that in this image. So you also see some carbohydrate chains, and they can either be attached to a protein, and we call that a glycoprotein, or the carbohydrate chain can be attached to one of the phospholipids, and it's called a glycolipid. But all these carbohydrate chains on the outer surface of the cell function in cell recognition. They identify the cell to the immune system, so that your immune system won't attack your own body cells. So they are cell recognition structures, glycoproteins and glycolipids. And then you have these large purple protein molecules that you see, and they're just called membrane proteins, and they play roles, and they play different roles. Some can be enzymes, some can function in transport. And then you see cholesterol, and we're going to talk about the role that cholesterol plays, and also what it means when you have more unsaturated fatty acids forming the tails of the phospholipids versus when you have more saturated fatty acids, because that plays a role too. So there's a video on active transport that is completely in the wrong order. I would not watch this at this point in time. It goes with this chapter, but it doesn't go with what we're doing right now. Anyway, it's on slide 10. So membrane fluidity. So we want a fluid membrane. It needs to be fluid, but we don't need it to be too fluid because then we would melt, right? So it needs to be like a semi-solid, kind of solid, but not really. It needs to be, everything in the membrane needs to be able to move around, but it doesn't need to be too much of a fluid because, you know, like I say, it won't hold together. So this is just an experiment that was done. It's showing you the results of that experiment where a mouse cell and a human cell were allowed to fuse together in a test tube. And the hybrid cell that formed, and they could fuse together, by the way, because the phospholipid bilayer of a mouse cell is the same as the one for a human cell. So when they find each other in a beaker of water or a test tube of water, they're going to kind of like fuse, you know, they're going to move together and fuse into a larger cell. Because those phospholipids do that. I mean, phospholipids fuse together with other phospholipids when they come in contact. But then all of these pink and purple structures that you see are the membrane proteins. And so what they saw was that the membrane proteins were a mixture from both cells. So that let us know proteins move throughout the phospholipid. This was the experiment that showed us that the proteins actually move around through the phospholipid bilayer. Now, so what does it mean if the fatty acid tails are unsaturated or if they are saturated? All right, unsaturated fatty acids, we've learned already, are oils at room temperature, and saturated fatty acids are solids at room temperature. So we need to have a composition of unsaturated to saturated fatty acids that will enable the plasma membrane to be as fluid as we need it to be, but not too fluid. It has to be kind of solid, or it's going to fall apart, okay? So there are organisms that live in cold temperatures, and in cool temperatures, the plasma membrane will actually end up switching from a fluid state to a solid state. So organisms that live in cool temperatures, they will end up with more unsaturated fatty acids in their phospholipid bilayer, because that will make the membrane go back to a more fluid state, okay? Then if they live in a hot environment, they're going to have more saturated fatty acids than unsaturated. So the membrane won't be too fluid. So this is what the unsaturated fatty acid tails look like, and they're going to make the plasma membrane more fluid because they prevent the packing of the phospholipids. And then the saturated tails, more saturated tails, are going to make the membrane more viscous or thicker, more solid, because the saturated tails are able to pack together. So that is the role of saturated and unsaturated fatty acids. Now, what about cholesterol? Because you probably saw the little cholesterol molecule. There are cholesterol molecules embedded within our plasma membrane as well. And what happens with cholesterol is if the temperatures are high, if it's hot, which means our membranes are hot, our plasma membranes are hot, cholesterol is going to function to restrain the movement of phospholipids. So it's going to keep them from being too movable, too fluid. But if the temperature is low, if it's cold, then the cholesterol is going to prevent the tight packing that you see with those saturated fatty acids. It's going to prevent the tight packing of the phospholipids because it's kind of acts like a spacer, you know, it's in between them. So it helps to maintain the fluid nature of the plasma membrane by preventing the tight packing of the phospholipids. So basically cholesterol does one or the other depending on the temperature. If it's really hot, it's going to keep the phospholipids from moving too much. And if it's really cold, it's going to actually allow more fluidity because it's like a spacer. It gets in between the phospholipids and separates them so they can move, so they can have more movement. All right. All right, this page just tells us that some of the membranes are peripheral. Some of the proteins are peripheral. Some of the membrane proteins are peripheral. That means they're on the surface, the outside surface of the cell. Some are integral. That means they penetrate into the core of the phospholipids. And then some span the entire plasma membrane. They are transmembrane proteins, and that means that they are large enough that they are found on the outside all the way to the inside of the plasma membrane. This is a transmembrane protein right here. Okay, so we have about 6 different types of transmembrane proteins, and they have different functions, and we're going to talk about each one. So some membrane proteins function in transport, and they can either be called channel proteins, like this one on the left. It just has a pore or a channel, and molecules can move across the membrane through that channel. Some membrane proteins, we call them carrier proteins because they don't have a channel, but they actually chemically bind to molecules, and then they change shape and open up and move them across the membrane. So a channel protein just has an open channel, and a carrier protein has to chemically bind and transport the substance across. And we have 2 types of carrier proteins, one that requires ATP, and we would call that a pump, and one that doesn't require ATP, and it's just called a regular carrier protein. There are also membrane proteins called enzymes, and they're just like any other enzyme. They catalyze or speed up chemical reactions. We have receptor proteins, and that a signaling molecule has to bind to that receptor protein, and then it will initiate some type of change inside the cell. So, for example, if this signaling molecule were insulin, and this cell were a liver cell, a human liver cell, then when the insulin binds to the receptor protein here, it will cause this liver cell to move glucose, to take glucose from your blood and store it. So it lowers blood glucose that way. We have glycoproteins, which have carbohydrate chains attached, and they function in cell recognition. We have joining proteins. We had all different ones. We had the tight junctions, gap junctions, desmosomes, but they attach cells to one another. And then we have proteins that contain, this is called fibronectin right here. They contain a substance that attaches to the extracellular matrix outside the cell and also to the cytoskeleton. So it attaches the plasma membrane, it kind of holds the plasma membrane in place. All right, and this is functions of the plasma membrane, and that's on slide 28. This is an example of basically HIV, causes AIDS. HIV is the human immunodeficiency virus, and it causes the disease called AIDS. Well, this is a cell or a vesicle that contains the HIV, or the virus, inside of the vesicle. And so the membrane on the outside is a phospholipid bilayer, and these little spikes on the outside are glycoproteins. So these glycoproteins can attach to receptor proteins on our immune cells called helper T cells. So basically go back to that receptor protein. Okay, so instead of a signaling molecule, it's a glycoprotein spike that's going to attach to a receptor protein on the helper T cell. Then it's going to trick the helper T cell into engulfing and making more copies of that virus. And it's because the helper T cell has these receptor proteins called CD4 receptors and these co-receptors called CCR5. So as long as the glycoproteins in the envelope here that surrounds the HIV, as long as they can attach to both the CD4 receptor and then the CCR5 co-receptor, they have to attach at the same time. Then the next thing that's going to happen is it's going to trick that helper T cell into engulfing that virus, making more copies of it, and then releasing more copies to go attack more cells. And it will eventually shut down your immune system and cause you to develop AIDS. The molecules that function in cell recognition are glycolipids and glycoproteins. So glycoproteins would be one of our membrane proteins. So cell identification, tissue compatibility, that's what cell recognition is. This slide is just showing you how the glycolipids and glycoproteins are actually produced. The glycolipids are produced in the smooth ER, and the glycoproteins are produced in the rough ER. And they are transported to the Golgi, and then the Golgi can package them in vesicles, and they can be sent to the surface of your plasma membrane of your cells, and produce that, show that carbohydrate chain on the surface, which identifies your cell to the immune system. So that's how it works. That is another example of the endomembrane system. Plasma membranes have selective permeability. They allow some substances to cross, and they don't allow other substances to cross. So we're going to talk about different substances and how they cross through the plasma membrane. The first thing we have to know is whether or not the substance that needs to cross is a nonpolar molecule. If it's nonpolar, and lipids, all lipids are nonpolar. So if it's a lipid substance, it's nonpolar. Then it's also, that makes it hydrophobic or insoluble in water. So these substances can actually pass across the membrane directly through the phospholipid bilayer. They can actually move through the phospholipids by diffusion, and they can move very fast through the phospholipid bilayer. So this is going to be hydrocarbons like fatty acids, amino acids, not amino acids, they're polar. Fatty acids, which are lipids, and they're composed of a hydrocarbon chain. And then the gases, carbon dioxide and oxygen. All of these lipids and carbon dioxide and oxygen easily pass through the phospholipid bilayer. But then you have polar molecules and molecules that have a charge, which would be ions. They can have a positive or negative charge. So polar molecules like sugars and amino acids, and also charged particles like ions, like a sodium ion has a plus charge, a calcium ion has a +2 charge. Any of those charged atoms, which we call ions, plus if they're polar, like sugars and amino acids, they are going to have to go through, they're going to have to either go through a channel or a carrier protein. And water is polar, so water goes through a special channel protein called an aquaporin. But water is also small, so water can pass through the phospholipid bilayer to a certain extent. So it's a little bit of an exception. But anyway, substances that are hydrophilic, that means they're soluble in water. So they're not lipids or carbon dioxide or oxygen. They're hydrophilic, they're soluble in water, they're either polar or they have a charge. They need a transport protein. Okay, they can't come through just the phospholipids. Channel proteins, have a channel that passes all the way through the membrane, and carrier proteins have to chemically combine with the molecule and then change shape and shuttle them or transport them across the membrane. There are special channel proteins called aquaporins that are only for water. And there's a video on aquaporins and the movement of water across the membrane, and this is slide 39. And then there are transport proteins for specific substances. For example, there's glucose carrier proteins. And this is Bioflix animation on membrane transport, and it's slide 41. And then we're going to talk about passive and active transport beginning in the next recording. So this is slide 42, and this is where we'll start the next recording on passive transport across the plasma membrane.